Hi everyone, and welcome to Results Day. So you hypothetically made your UV plate. So you streaked bacteria horizontally on a plate. You covered half of it with foil and exposed to UV light. And you came back to lab, and this is what we saw. So each of you streaked different bacteria, and we did different UV exposures, and this is the layout. So let's walk through how to read or score these results. So I'm going to refer to this example plate up here. So in this example plate, the side that was covered by the foil is which side? The left or the right? The side covered by the foil, hopefully you said the left, all right, is the side that was not exposed to UV. And so that side should grow just fine. That is our control side, okay? The other side is the side that was exposed to the UV, and presumably we should see some effects of that UV light. We should see some reduction of growth. So if we don't see any reduction of growth, if we saw the same amount of growth on both sides of the plate, if both sides look the same, all right, then we would score it with 4 plus marks. Basically, we'd say there's a lot of growth. If we didn't see any growth, if this whole side of this plate is clear, then we would score it with a negative sign or a 0, either one, to indicate there is no growth. And then in between, for everything in between, we can give it a relative score. So how we're scoring this is relative to the control side of the plate. So I'm scoring the growth on this side of the plate by comparing it to the growth on this side of the plate. So this is pretty thick on the left side of the plate. I mean, you can't see any colonies. This is just lawn in the shape of the streak line. And then on the right side, I can see a handful of colonies, maybe like 20 or 30 colonies. I'm not going to try to count them. I'm just going to say, about what percent do I think? And I think that this is maybe 5 or 10% tops of what's growing on this side of the plate. And that's a real rough estimate, but that definitely falls into the slight growth category. So I would score this plate, if I was scoring it, as with a 1 plus sign, okay? And so you're going to go through and score all of these plates. So what we have here, the way I've displayed this is, I laid out all the plates. On top, the top row of plates are all plates that had Bacillus subtilis on it and that students had streaked. And then on the bottom row, these plates all have E. coli growing on them. And the first plate was exposed to UV for 10 seconds, the second plate for 20, and so on and so forth down the line. It's always the right side of the plate that is the UV exposed side, and the left side of the plate is the control side. And some things I'll point out that you'll notice, like on some of the plates, like on this plate right here. So here's the line they drew. But you'll notice that even on the left side of this line, the control side of the line, there's blank spots where no bacteria grew. And that's probably just bleed through from the UV light. So it might have had to do with how I angled the light. If I shown it at an angle, it may have actually gone underneath the foil a little bit. So that's why it's not grown perfectly right up into the line. Yeah, that's it. And some of these plates, like this one right here, okay, not a lot of growth over here. I would expect to see lines, stripes of growth. I think that this might have been due to some technique errors that this student might have had. You'll notice that the bacillus colonies grow really fat. Like this is a bacillus colony right here, a single one, versus like here's a little E. coli colony right here, like a tiny one. Okay, so they definitely have very different colony morphologies. But again, we're always comparing, when we're scoring, we're comparing the growth on the right side to the growth on the left side of that plate. So let's just do a couple. Let's do this first one here. together. So this is the bacillus at 10 seconds, so I'm going to record it in this box here. If I look at the two sides of the plate, they don't look significantly different to me. It looks like the same amount of bacteria. It also looks like the students streaked the plate the wrong way. They were supposed to make lines this way through the perpendicular line drawn on the bottom of the plate, not parallel to that line, which is like what it looks like it did based on how the bacteria grew in those vertical stripes instead of horizontal stripes. But anyway, I would still say there was no reduction of growth. So I'm going to give it 4+ marks. All right, versus the E. coli at 10 seconds, there's definitely a lot of growth on this foil covered side. And there's definitely less growth on the exposed side. So the question is, how much less growth? I think it's definitely less than 30%. So I'm going to say it's slight growth. So I'll just give it 1+. And then so on and so forth. You would continue down the line and score each of these plates. All right, and then on the next page of the results, so you'll fill this out on paper. And if you can print it, ideally you print it and you fill it out in this chart. If you can't print or you don't want to, then just draw this chart on a piece of paper and write out your scoring. And then you're going to graph your results. So on this graph, the y-axis, I have the amount of growth, and on the x-axis here, I have UV exposure time. So I didn't label this axis down here, so you should do that. Indicate the times. It's okay if it's not to scale, so you can just do 10 seconds, 20 seconds, 30 seconds, so on. And then you're going to do a line graph. So it's not a bar graph. I don't want to see a histogram. I don't want to see bars. I want to see dots at each point, and then the dots connected by lines. That's a line graph. So you do need to have a key. You should have 2 lines, really, because you're going to graph the data for the bacillus, and you're going to graph the data for the E. coli. And so there should be 2 lines. Please make them 2 different colors or 2 different patterns so that you can tell which is which. And you'll need to make a key. So like maybe right over here, oops, right over here you might write your key that the bacillus is blue and E. coli is green. You can do it by hand on this sheet, or if you want, you can transfer this chart you can do it in Excel. So it looks all pretty and to scale, but that's not required. So either way, you can do it by hand or by computer. And then don't forget to answer. That was weird. I think I picked the drawing tool and paused the recording. So what I was saying was don't forget to answer this question at the end. This is important too. So based on your results, which organism would you say was more resistant to the UV light? And why do you think that is, based on what you know about that bacteria from the lab intro? All right, that's all. And I have to get to a faculty meeting. How do I stop?